everyone joining today, and thanks for tuning in. I am Isabela, a Master's in Movement Science at Unesp, Brazil, and the virtual intern of the Exercise Physiology and Cardiovascular Health Laboratory at the University of Ottawa Heart Institute. I'll be your host for today's special episode of BEaTS Research Radio, a YouTube channel and podcast which aims to disseminate science to the community. Our guest today is Dr. Leila Mostaço-Guidolin. Dr. Leila received her bachelor's degree from the University of São Paulo, Brazil in 2007. At the same university, she obtained her master's degree in 2009 in physics applied to medicine and biology, whereas her research project was focused on biomechanical characterization of health and neoplastic cells through vibrational spectroscopy. After that, Dr. Leila in 2015 earned her PhD at the University of Manitoba, Canada, with her thesis investigating the development and application of image analysis methods to monitor the progression of atherosclerosis. Currently, she is an assistant professor at the Carleton University with the Department of Systems and Computer Engineering. Her research interests are primarily in the areas of image analysis, extracellular matrix remodeling, and 3D bioprinting. Welcome, Dr. Leila.
Thank you very much. It's my pleasure to be here today.
First question. Your research lab, the CUPI, focus on developing tools that are useful in answering relevant biological questions related to the assessment of tissue function during normal and disease process. Could you please tell us some tools that are currently being developed by your group?
Sure. Yeah, so in my group, we have now quite a bit of projects going on. Some are in the beginning, some are a little more advanced, but all of them having as a common theme the characterization of the extracellular matrix. And in our lab, we use several tools, and one of the main ones is called nonlinear optical microscopy. So that's a special type of microscopy where we can basically see the structures of biological tissue without the need of any preparation, any labeling, any additional chemicals in there. So we can get signal, get information about these structures just based on the composition of the tissues. And in biological research, that's something quite amazing. We can get information about, for example, collagen fibers, elastic fibers. We can look at structures that are rich in lipids. And as I said before, in my lab, we are interested in looking at how the structure of the extracellular matrix plays a role in terms of how they affect cellular responses, how they will change the inflammatory responses that we have in tissue. And we are very interested in characterizing collagen per se, so how the collagen fiber changes may affect the whole environment that we have in tissues and cells. And our goal is to understand better what's going on related to fibrosis. So fibrosis is basically an accumulation of collagen when we have way too much collagen in the tissue. And that triggers quite a bit of different responses. The cells start to behave in a different way. So we have inflammatory responses happening there as well. And it's interesting because that's a condition that's happening in different diseases, from cancer to asthma, for example. And nobody understands very well how do we connect fibrosis with inflammatory responses and so on. So we are trying to help a little bit to understand those mechanisms.
Thank you for elucidating this information, Dr. Leila. Our second question, diving in your lab projects, the bioengineer artificial lungs development models to recapitulate pulmonary fibrosis, where your group is developing models to be used to understand the mechanism involved in pulmonary fibrosis. What are the advancements of this project?
Okay, yeah, so this is a very interesting project, and this one is going to take a little while still. And we are working with, in collaboration with researchers from the National Research Council here in Canada. And the idea is to develop models or structures that we are going to replicate the structure that we have in airways. So for example, we are going to have the interface between air and liquid in there. The plan is to build or bioprint those tubular structures where we have air being perfused inside of the tubes, And then we can allow the cells to differentiate and to behave a little closer as they would in normal tissue or in vivo. And also on the external side of these tubes, the plan is to have different types of cells and arrange the matrix, extracellular matrix, in a way that we can build the structure that we would expect to see in a normal airway. And the idea of this research in the future is to have these models functional enough for us to be able to explore and assess how well some therapies or some new drugs are doing in terms of addressing the remodeling that happens in the tissue and how the cells are behaving and so on. And hopefully we can understand a little more all the details and all the mechanisms that are happening there.
Oh, perfect. Thank you, Dr. Leila. One of your latest articles published at the important American Journal of Respiratory and Critical Care Medicine investigated how a defective collagen organization contributes to airway remodeling in asthma. What do you consider as the main result of the study? Could you please point out the clinical relevance for the treatment of respiratory disease?
Sure. Yeah, this was a very interesting work because we were able to show collagen fibers and the remodeling that happens in airways with a level of detail that was not seen before. So before, many of the studies, they were all relying on histological images. So it was possible to see that there was a lot of collagen being accumulated in the airway walls. But up to that point, nobody could have seen in terms of at the single fiber level, the details and how they were being organized in there. So with this work, we managed to share or to see that the collagen fibers in asthma, asthmatic airways, they are actually more disorganized than in normal. And that led us to start to investigating more what was behind these changes, right, in asthma. So making a very long story short, we eventually found out that decorin, which is a special type of protein responsible for the fibril arrangement inside of the collagen fibers, was expressed or in lower levels in asthmatic cells. And therefore, it is one of the possible causes of this remodeling that we observe in collagen. And that was very new information that we managed to publish and to share with the community, the scientific community. And we are expecting now, and we are seeing that this piece of information is now triggering different works and different research related to exploring how we can improve or how we can come up with different therapies to address asthma. Because right now, asthma is, unfortunately, there is no cure. So all the medications that we have available, they all address the symptoms, but we don't understand yet what's behind all this remodeling and all these effects related to the inflammation and so on. So hopefully we were able to place a little more information into this big puzzle that there are so many people working on, and who knows, maybe we are going to get a little closer to developing a therapy, right, for this disease.
Yeah, thank you, Dr. Leila, and congratulations for your findings, important findings. So our last question today, considering your international background and your personal experience as a previous student in Brazil and currently a successful international professor in Canada, do you have any advice for women scientists who are just starting their careers?
Okay, that's a good one. I would say be aware of opportunities and be curious, and don't be afraid to take chances or take risks. Don't be afraid of changes, be that changing area of research or moving from one city to another, from one country to another, and do your best to adapt. We learn a lot of things through the years, and we change quite a bit as well. We get more information, we become more mature, and that's normal. It's important, of course, to have goals, to know more or less what you want to do, but don't be too concerned or afraid if those goals change or if you have to adapt a little bit, because that's probably the fun part of life, right? So even myself, if you had asked me where I would be when I was finishing my undergraduate, I would never have guessed that I would be here today. So, and I don't know what I will be in 15, 20 years. I have a plan, I have an idea, but who knows what's going to happen between now and then, right? So take it, take the opportunities, chat with people, adapt, and things will happen. Just don't be afraid.
Yeah, perfect. Thank you for your brilliant advice. So this concludes our interview. I would like to thank you, Dr. Leila Mostaço-Guidolin, for the fascinating talk with us regarding bioengineering research. Thanks to our listening for checking out this BEaTS Research Radio episode. Subscribe to our YouTube channel or find us in your favorite podcast app. On behalf of the whole team here at the BEaTS Research Radio, stay safe and heart health.